So every single last one of us has been impacted by cancer in some way, shape, or form. And one of the most common questions that is raised around cancer is there a cure? And is the government hiding it from us? And if there's not a cure, why not? Because we've dedicated trillions of dollars and decades of research. There should be a cure by now, right? Well, I'm a cancer biologist. I've been in the industry for about 12 years, and I am here to tell you there's not a cure for cancer. And the reason's actually really interesting and kind of simple, but also kind of complex. And it's going to bring us into a journey into molecular biology and how cancer works. And it's actually very interesting. So if that sounds good to you, let's jump into it. So first of all, what exactly is cancer, generally speaking? Well, most people know that it's the uncontrolled proliferation or division or growth of cells. And this can be any cell in our body. Well, I'll caveat that. Any cell besides red blood cells, platelets, and germ cells, like sperms and eggs. But depending on where the cell of origin was, like what cell became cancerous, defines like the general class or type of cancer. So things like skin cancer, lung cancer, blood cancer, brain cancer, breast cancer, digestive tract cancer, all kinds of different types of cancer. And it all depends on where the cell was that became cancerous. But at the end of the day, they're all the exact same thing, generally speaking. It's just the uncontrolled proliferation of some type of cell in our body. And left unchecked, if it's a solid tumor, it will just grow and grow and grow and spread all over the place without treatment. Treatment can slow it down, sometimes stop it. And in the blood, it can crowd out the bloodstream and, cause problems that way, and the same consequences can happen. If it's all the same kind of thing, though, then why can't we just stop the proliferation of the cells and have a general universal cure for cancer? Well, the answer to that comes from the actual genetics of the cells themselves. So here's where it gets really interesting. So if you look at a cell, okay, every cell in our body has a bunch of DNA in it. And in fact, every cell in your body has the exact same, theoretically speaking, sequence of DNA in every single one of your cells. And we've got about 3 billion base pairs in our genome, which the genome is just the entirety of your DNA. And if you took one single chromosome and stretched it way out and you zoomed way in on it, eventually you would get to this point right here where you have base pairs lined up in a row, just like this. And at certain points throughout the chromosome, you would come across things known as genes. And they look the exact same as the rest of the DNA, but genes are special because genes are used to code for proteins. And proteins actually go and do stuff. They're the functional units in our cells. The DNA doesn't do anything. The proteins are the ones that kind of float around and have functions. But the DNA, the sequence of the DNA encodes for the sequence of the amino acids of the proteins, and then that sequence of amino acids dictates the function, the folding and the function of the protein. This is where cancer kind of starts. So let's imagine that this pretty little purple and yellow protein here is involved in cell division. And there are plenty of proteins in our body that perform this general function. And if the cell wants to divide, it will have this protein activated. And if not, it will inhibit this protein. And there's plenty of ways that the cell can do that. Like look up phosphorylation is one way that the cell can change the function of a protein. So with this cell inhibited, then cell division is suppressed, if not completely impossible. And this is fine. But let's now imagine that this sequence of DNA that encodes for this protein acquires a mutation right here. And mutations can be caused by all kinds of different things like tobacco and smoke, UV radiation from like the sun or tanning beds, and all kinds of different sources. And let's say that this mutation right here is actually at this C right here, and it changes the C to a T. Seems totally harmless, right? And usually mutations are completely harmless. They don't do anything. But sometimes you get a mutation right in the gene, and the mutation is unfortunate enough to actually cause a structural change to the protein that it encodes for. And sometimes those structural changes can actually lead to significant functional changes. Now let's imagine that this C to T mutation results in this protein no longer being able to be inhibited. So now the cell is like, hey, we don't want to divide, but this protein can no longer like hear the cell. It's always on, always active, and always promoting cell division. And now if this protein is that significant in cell division, you can have uncontrolled proliferation of this cell from this single mutation and this single mutant protein. This is the general kind of theme of cancer, but in reality, it's usually multiple different proteins. You need like kind of a cascade or an array of different mutated proteins that leads to the uncontrolled proliferation. And it normally isn't just one mutant protein. But this is the general idea. Mutant proteins can result in uncontrolled proliferation, which leads to cancer at the macro scale. So if you clean this up, this equation looks pretty easy to inhibit still. You get mutations that lead to mutant proteins that lead to uncontrolled proliferation. Let's just develop therapeutics that inhibit these proteins, because that should inhibit the proliferation of those cells. This is basically what targeted therapeutics is, right? We can examine the biology of a particular type of cancer. Let's use breast cancer, for example, like HER2 positive breast cancer. You can examine the biology of like a big swath of HER2 positive breast cancers and say, hey, it looks like in 83% of these cancers, we get this like signaling cascade is very highly active. Let's try and inhibit it. That's targeted therapeutics. The issue is that it becomes very complicated when you look at cancer as a whole, because HER2 positive breast cancer is way different than lung cancer, which is different than skin cancer. And they have different types of proteins that tend to lead to the uncontrolled proliferation of those specific kinds of cells. So we're already looking at no longer like a universal general cure for all cancers, because cancer really isn't even a single disease. It's a category of diseases. And even within the same type of cancer, like this is melanoma, superficial melanoma, even two people with the same cancer tend to have different genetic mutations that result in those cancers. And cancer cells are very unstable. So as the cancer grows and over time, it can accumulate more and more mutations. And as the disease progresses, it just becomes more and more and more unique, to the point where you can literally have two people with the exact same disease, give them the same therapy, and one person can become cured and the other person doesn't even respond. And this happens a lot like in clinical trials. And a lot of people have problems with clinical trials because it's like, well, this treatment apparently is super good for this kind of cancer. And my dad was in that clinical trial and he didn't respond at all. And it's like, that just happens a lot. And it's because cancer is just really complicated. And it's because genetics is really complicated. And it's basically individualized, essentially. And not to mention the fact that this situation where we have an actual targeted therapy targeting a mutant protein for a certain kind of cancer is generally not the case, because this takes a lot of research time and money to figure out what exactly is the mutant protein we can target and how do we develop a therapeutic that can efficiently inhibit that mutant protein. It's really hard. So most cancer therapy doesn't even do this. It isn't targeted to a specific protein. Well, it is, but it isn't like a disease-specific protein. Normally, anti-cancer therapy just attacks cell division, because cancer cells tend to divide very quickly. So if we can inhibit cell division, we can inhibit them from dividing. But the thing is like, this is a very mediocre strategy for many reasons, one of which is that you can't use very high doses of these kinds of therapeutics because other cells in our body divide, like healthy, necessary cells also divide. And it's why even people who are getting just like general chemotherapy experience a lot of toxicities, because it's going to kill a lot of healthy tissue as well. So this is why there's not a cure for cancer. It's just really complicated. But hope is not lost, because there are other kinds of therapeutics that are being developed nowadays that are more general in their activity, and there's general therapeutics that lead to personalized outcomes, which is the goal. If cancer is personalized, your disease is your unique cancer. But we're looking for a general kind of therapeutic. We need to develop therapeutics that are general in their basic mechanism, but actually act differently in each individual person. But how do you do that? Well, one area is this. Every single cancer, with some exceptions, in order to survive, has to inhibit the person's immune system, because the immune system is actually very, very good at finding and killing cancer cells. You might be thinking, well, if it was that good, then how do people get cancer? Exactly. In order for cancers to live, they have to actively suppress the immune system. So you can develop therapeutics called immunotherapy that are targeted to the immune system that are meant to either suppress the suppression and unleash the immune system, or are actively meant to stimulate the immune system against the cancer. And since the immune system is personalized to the individual people, right? Like Tim's immune system is different than Fred's immune system. And even though Tim's cancer and Fred's cancer are very different, Tim's immune system is very good at finding and killing Tim's cancer. But Tim's immune system is just suppressed. So if you can activate it and stimulate it against his cancer using some general mechanism, you can actually have a general therapeutic that leads to personalized mechanisms through immunotherapy. And that's just, and again, immunotherapy is a general class. There are different kinds of immunotherapy. Like, for example, cancer vaccines are considered immunotherapy. But this is just one of the general ways that scientists are using nowadays to improve anti-cancer therapy. And the future looks very bright. Now, Bio to Bedside, this channel, is going to be dedicated to talking about all of the new kinds of cancer therapies, explaining cancer immunology, and just kind of doing like Khan Academy style videos to explain the relatively intricate biological mechanisms of cancer and new cutting edge cancer therapeutics. So if that sounds interesting, subscribe, hit the bell, do the whole thing. My name's Jeff Martin. I'm one of the co-founders over here at Bio to Bedside. Hopefully you enjoyed this. And yeah, all right. I'll see you in the next video.